Jean-Baptiste, you're muted.
Hello, everybody. I'm Jean-Baptiste Born, and I'm the Attaché for Science and Technology at the French Embassy in the US. I would like to thank all our panelists for being connected to this talk entitled There is a Pattern, Identifying Common Biomolecular Mechanisms to Cancer, Infection, and Neurodegeneration, and congratulate the UCSF QBI for their fantastic organization of this event. Many thanks to Jacqueline, Fabius for the moderation of the incoming panel. As far as today's panel is concerned, let's remind us that recent technological advances in several fields outside of biology, such as physics and computer science, make it possible to analyze biological systems using quantitative methods. For example, the efforts of biomedical researchers to understand how genes work together to produce RNA that codes for proteins that make up cells, tissues, and organs can be augmented today by the skills and different approaches of engineers, mathematicians, physicists, and computational scientists, just to say a few. This multidisciplinary approach was at the heart of the collaboration between QBI and Pasteur that told us so much about the SARS-CoV-2, but the promises of this approach extend far beyond virology and infectious diseases. So how we can break the walls between all disciplines and devise patterns and common systems through quantitative analysis in order to understand better biomolecular mechanisms is what this panel is going to talk about. So now we will switch to the introduction of our panelists, starting with Jacqueline.
Good morning, I'm Jacqueline Fabius. I'm the Chief Operating Officer of QBI, the Quantitative Biosciences Institute.
Good morning, I'm Nevan Krogan. I'm a professor and director of the Quantitative Biosciences Institute, and my research focuses on developing technology in an integrative way so that we can study the underlying biology across many different disease areas, including cancer, neuropsychiatric disorders, and infectious disease.
So good morning, I'm Christophe. I work at Institut Pasteur in Paris, France. I'm the scientific director, but also the head of the Fungal Biology and Pathogenicity unit, which focuses on fungal pathogens of humans.
Hi, my name is Shaeri Mukherjee. I'm an associate professor in the Microbiology and Immunology department at UCSF. And my lab studies how pathogens can be used as tools to understand basic principles of cell biology.
Hello, my name is Bruno Goud. So I am working in Paris. I am a cell biologist and research director at the CNRS and Institut Curie. And I am the chairman of a program that support interdisciplinary research and gather several institutes in central Paris.
So good morning, Nevan and Shaeri. Good evening, Christophe and Bruno. Thank you everyone for joining us. It is my pleasure to moderate this really fantastic panel. There's a pattern, identifying common biomolecular mechanisms to cancer, infection, and neurodegeneration. During this hour, we'll be discussing commonalities across disease states, and later in our discussion, we will also be discussing the importance of collaborations in science. So I'll start with a question. Could you talk about certain disease states that cause or connected to other disease states? Nevan, perhaps would you like to start?
Sure. There are certain diseases that can cause or are connected to other disease areas that are known. I think there are many that are unknown, but there are some that are known, specifically when you're looking at viral infections. So, for example, HPV, that is an infection connected to a number of cancers, including cervical cancers. I think pretty much 100% of cervical cancers are connected to HPV. More recently, the majority of actually head and neck cancers are connected to HPV as well. Now that smoking is on the decline, about 60% of the head and neck cancers that are seen are actually now connected to HPV infection. Other viruses that are connected to cancer include hepatitis B and hepatitis C and liver cancer. I think the majority of liver cancers are now being connected back to hepatitis C. There are other viruses like EBV and certain cancers like stomach cancers. So those are more direct. And of course, there's some indirect connections, for example, with HIV infections, with Kaposi sarcoma, that's kind of a secondary a disease that happens when the immune system is compromised. So there are a number there of viruses connected to cancers, maybe half a dozen or so. But as more work gets carried out, I do believe there'll be more disease that will ultimately be connected to viral infections. We just don't know about it. And I think that's a topic that could be discussed today.
So following the topic of cancer, could you discuss how research on cancer is providing opportunities for looking at new antifungal approaches? For example, drugs targeting bromodomain proteins or targeting kinases. Christophe, would you like to take this one perhaps?
Yeah, so what you should realize is that a number of pathogens are eukaryotes. So in their organization, they do not really differ of the organization of our cells. So components of eukaryotic cells, such as yeast or parasites, are not very different from components of mammalian cells. And so in the field of cancer, there are a lot of research that has been carried to identify drugs that could prevent cancer. They could target protein kinases, they could target these bromodomain proteins, etc. And so what we start to realize is that these compounds can also be effective against eukaryotic pathogens such as yeast or parasites. And so there are, for instance, examples in work developed by the group of Leah Cohen in Toronto, Canada, that shows that one anti-cancer compound targeting a protein kinase can be used to actually treat fungal infections. And there are other examples such like these. So I think the wealth of compounds that have been developed in the frame of cancer research will have impact beyond that, and especially in the field of infections.
Thank you. And Shaeri, I feel like you had something to say about this as well.
I had a thing to add to what Nevan said about the relationship of viruses with cancer, and I think we can add bacteria to it as well, especially H. pylori, and Nobel Prize on H. pylori for discovering that it causes cancer. So I think many pathogens have maybe we have not yet found all the links yet, but it's a really exciting area of research.
Yeah, bringing that up.
Yeah, I think I don't know the exact number, but it seems that about 20% of all cancer are caused by viruses or bacteria. So it's a huge number actually.
Yeah, if I may add, because I'm a fungal biologist, but there was a recent paper in Nature showing that liver cancer or pancreatic cancer as a trigger in the microbiome. And so this is influenced by the presence of specific fungi in our microbiota. So I think this goes beyond viruses and bacteria, and also include fungi and probably parasites, because that not in humans, but in cattle, Stellaria is responsible for cancer as well.
Thank you. So Nevan, perhaps we would touch on this a bit later, but I believe that you've also found some connections with kinases and COVID-19.
Yeah, we have globally looked at which kinases are being hijacked during the course of SARS-CoV-2 infection. And there's like 500 kinases in total. We identified about 10% of them that are seemingly being hijacked. And that's important because many of them are druggable, and many of them have drugs against them, and they're being used a lot as anti-cancer agents. So through drug repurposing, we found that a number of these anti-cancer drugs that have been targeting in different kinases that seemingly the virus needs to infect our cells have potent antiviral effects. So the fact that you do see this crosstalk across these diseases allows one to, through drug repurposing, determine if maybe a drug for one disease could be used for another one. It could be even more effective on another disease, which I think is, again, a theme of today's discussion is how these commonalities can actually lead to therapeutic insight.
So just taking a little bit of a turn, there's an interesting article in Nature yesterday morning making a connection between dementia, specifically Alzheimer's, and infection. So how strong is the evidence that some forms of dementia, such as Alzheimer's, are caused by an infection? Bruno, would you like to chime in?
Yeah, I have read this paper also, and I noticed that someone in the state gave $1 billion or $1 million to, if you can prove that Alzheimer as it's originate from bacteria. Yeah, so I think so. It's not my field, but I think it's an interesting field. I mean, there have been several papers showing that there is a close link between infection and Alzheimer, but I think that more recently, I think that some paper appears saying that it could be a valid hypothesis. And for instance, I have seen a recent paper also in Nature from a group in St. Catherine, showing that infection of glial cells can stimulate gamma secretase activity and the formation of the amyloid plaque. So I think it's an interesting field that makes the link between infection and Alzheimer. Even so, I think most of the people of the community still do not believe that Alzheimer is caused by infection.
Thank you. How have pathogen studies led to the identification of important cellular pathways or other disease states? Shaeri?
Yeah, so as a cell biologist, I've always, wondered and have been amazed as to how a single cell organism can kill a 40 trillion cell human being, and especially like viruses with only a few genes. And I think the reason that happens, and the fact that we as humans need antibiotics or vaccines or other things to protect ourselves, is because that these pathogens have really understood the inner workings of our cell. And by studying these pathogens, we can sometimes uncover things that we otherwise didn't know. And a prime example that comes to mind is from work done at UCSF here. So it was known that this virus Rous sarcoma caused a disease in chicken. It was named after Peyton Rous, who discovered that. And then our former chancellor at UCSF, Mike Bishop, started studying that Rous sarcoma virus, and he discovered that the gene that encoded the information to cause cancer was this gene which he called VSARC, or virus sarcoma, right? And then he looked to see homologous genes in humans, and he found the CSARC, which he called the cellular SARC, and that indeed was the first oncogene ever identified and led to the understanding that human genes can get mutated and cause cancer. Before his discovery, people thought that cancers were caused only by viruses and bacteria. And by just studying the virus itself and understanding how it works, he discovered the first oncogene. And so I am very, very much influenced by him. And my lab is next to his lab. He has since retired, but I find it fascinating how studies on bacteria and viruses have led to major discoveries. And it doesn't end there. Think about restriction factors, think about CRISPR, think about reverse transcriptase. All of these have come out of studies of just understanding how a virus or a bacteria works. So yeah, it makes me very excited.
Go ahead, Nevan.
I was just going to say, just to add to Shaeri's comment, we study a lot of viruses and bacterial species, and one of our goals is to identify biology that could lead to something therapeutic. But I actually think of these as the viral proteins and the bacterial proteins as probes. If you just want to understand important mammalian molecular biology, you put them in the cell, you pull them out, you see who they're talking to, you're seeing which pathways they're disrupting. You let the pathogen tell you what's important, because they're a lot smarter than us. So I couldn't agree more, and I'm very excited too, as using viruses and in particular and bacteria to just tell me what to study, tell me what to focus on. regardless of what it's doing, because it's going to be spending its time hijacking key molecular machinery.
Yeah, and also use them as cell biology tools, right? Like so many, I'm sure Bruno will agree, like VSVG studies have really uncovered how vesicle trafficking works inside the cell. And the reason that is because, and I can, speak of like from Legionella, examples on Legionella, which is not considered a huge pathogen burden. But what, Legionella has shown is that, some of these bacterial proteins, viral proteins, I'm sure fungal proteins as well, because they have limited time in the host cell, they know to target the key host protein, to sort of make them, achieve their replicative niche. When they're, sort of facing a lot of attack from the host, they have to kind of evade that and then make their home in a very toxic environment. So because of that, like if you compare the enzymes of the bacteria with the host enzymes, you will oftentimes see that the bacterial enzymes are 1,000-fold more efficient, or has a higher activity, and by using that you can use those as also cell biology tools to probe these individual functions.
Before I move sorry, go ahead, Bruno.
Another example, it's my field is the tool that for instance, tetanus toxin or botulinum toxin that cleave the snare protein. They have been key to identify, the mechanism of fusion of vesicle with target membrane. Nobel Prize of Jim Rotman, for instance, a few years ago.
So we have
led to the discovery of Botox, right? Like to help people, old rich people get an advantage too.
I think more than just the old rich people. So we seem to have a very engaged audience. So we have a few questions coming in, and here's the first one, which goes out to any of you. Is there any common characteristics of the viral and/or bacterial or fungal infection when it causes cancer, or is it more dependent on the host or genetic background?
Yeah, maybe I could chime in. That's a very good question. If you look at certain cancers, when you look at cervical cancer, 99% of cervical cancer or more is attributable to HPV. Okay, so that's being driven a lot by the virus compared to head and neck cancer, where maybe it's about 50-50. So I think in certain cases, a lot of it is dependent on the host, not just on the virus. The virus comes in, using HPV as example, and hijacks certain cancer genes like P53 or RB, and oftentimes that results in cancer, but doesn't result in cancer every time. So why not? So there's something about the host that is allowing one to fight off the infection more effectively, or if these proteins are being hijacked by HPV, it's not causing cancer. There's other mechanisms in place, or maybe there's variants within these cancer genes that are making them resistant to the virus, resulting in cancer. So I think it's a great question. You don't just look at the pathogen. It's a two-way street. Understanding the host is crucial, and looking at the host pathogen interface. And a lot of work that's being done by us and others is really trying to understand the host response to the pathogen, of why some people get infected and not, and then downstream why some people get downstream diseases and others don't. So it's a great way of looking at it. No, let's not just look at the virus, let's look at the host as well.
So Nevan, just to be clear for the audience, when you speak about the host, you mean the human body?
Yes, I'm talking about the human body, or if we're using other models like mice models, or just human cells, primary cells or cell lines, looking at host cells. But ultimately the goal is to have an impact in the host, which is us, which is obviously the human body.
What are some specific examples of common biomolecular mechanisms that are shared by multiple disease areas?
Well, I guess I alluded to a couple just now in terms of cancer-causing mutations. Probably the most prevalent gene that's mutated across all cancers is P53, or at least one of them. And there's a protein in HPV it's E6 or E7, I can't remember now, that specifically binds to and inhibits P53. So there's a great commonality there. Similar with HPV, there's another connection between another HPV protein and RB. This is another a protein or gene that's very highly mutated across many different disease areas. So there's in P21 and P16, and there's some other cancer genes that become mutated or get hijacked by HPV. So there's a lot of examples like that, specific examples of genes that can be linked to cancer, but then are also linked to certain infections like HPV.
I mean, there are also, other examples. I mean, one is, as common mechanism is, of course, the oxidative stress, which is very important for cancer, but also for other pathology like diabetes and neurodegenerative disease. So I think it's, and of course you have, also, in cancer, but also in other disease, all the alteration of the signaling pathway that are very important for many pathologies.
Shaeri?
Yeah, I wanted to add to that a little bit. Also, like in terms of Parkinson's, right? Like PINK and Parkin are genes that are linked to Parkinson's disease, which is a motor neurological disease. And recent studies have shown that these enzymes actually are involved in xenophagy or removal of pathogens from the cell via the overall autophagy pathway. And more people are showing that without those enzymes being functional, the defense against pathogen is also limited. So that's another common modality.
Could you illustrate perhaps the link? Sorry, go ahead.
Christophe, I think was going to add to this.
Yeah, so I wanted to add that with a number of intracellular bacteria, these bacteria are actually affecting a number of pathways that are also involved in cancer, for instance, and probably Shaeri can comment on that. But it's affecting signal transduction pathways. It's affecting mitochondrial function. It's affecting epigenetics, so the packing of the DNA and how the DNA is expressed. So bacteria, especially intracellular bacteria, have evolved a number of strategies to manipulate all cells and take advantage of the modification of pathways which are also modified in cancer or in other type of chronic disease.
Yeah, exactly. I think the reason that is because bacteria need a little more time in the host cell than a virus needs, and so it needs to replicate. And there are some bacteria that cannot replicate outside a cell, right? Like Chlamydia trachomatis. So what that means is that for them to survive inside the cell, they have to figure out what happens when the cell wants to undergo mitosis or cell division. And for that, chlamydia has been shown to bind to proteins of the centriole and sort of stop, the division, or in some cases do the opposite. And so as soon as you hit mitosis, right, that's like a hallmark of cancer, right? Like when there is a problem with cell division, obviously, a lot of things can go wrong. So, and then in terms of intracellular pathogens, I also think that because they have to go to a certain niche, it's like some bacteria go to the Golgi, some go to the ER, some go to the cytosol. What that means is they have to manipulate trafficking pathways, so how proteins travel. And all of these proteins are then linked to signal transduction pathways. So RAB are a common target of a lot of bacteria. And then again, RABs are needed for efficient membrane trafficking. And again, when you impinge on those processes, of course, that can have major consequences for the cell.
I have a question, which I think is going to lead into the great audience question we have below. So it seems that in all of your dialogue, cancer keeps coming up over and over again. And the question is, are there certain sets of diseases that are more correlative than others? Bruno, would you like to take a stab?
So cancer, of course, is it's correlative disease, but to be honest, I don't. So maybe Nevan, you can be better answer this question than me.
Sure. I mean, I would say, as we discussed, if you look at in certain infection states and the onset of cancer, I think that's highly correlative. And I guess going more broadly speaking, is it is that the most correlative set of diseases, or is it just that's where we've looked? As we discussed earlier, there's a big debate now about infection and neurodegenerative disease, and now that some of these unbiased tools are pointing in that direction, and that piece of nature that Jacqueline alluded to yesterday is doing just that. When we start to look in those directions, maybe we would see even tighter correlations with infection and other disease areas like neurodegenerative, maybe neuropsychiatric as well. So, I would say probably if you're looking broadly at different disease areas, I think infections and cancer are probably a pair that are most highly correlative, but it'll be very interesting to see as more unbiased approaches get employed to study different disease areas, which other ones are correlative. And I think you're going to see a lot more correlations across diseases when more data is collected.
So I think this leads well into our fantastic audience question from MB. Is biology becoming more and more like climatology, a science of systems in which no element can be studied in isolation? What provoked or allowed that change? Christophe, what are your thoughts?
Well, I think the change is coming in part from our ability to look at biological systems with high throughput technologies. So the so-called omics, so genomics, transcriptomics, proteomics. So technologies which allow you, for instance, for transcriptomics to look at all mRNAs into a cell at a given time in a given condition, or proteomics for proteins. And so I think this is allowing us to actually not look at a very specific thing, but really look at the cell at a broader scale. And you can apply that then, at the level of the host-pathogen interaction, for instance, as for instance, Nevan has been doing in the frame of COVID-19. But you can then look at the level of the tissue, or you can look at the level of the host. So these technology have really changed our way to look at biology, and I would agree that biology is really becoming more and more systems biology than it was maybe 20 years ago.
Bruno, do you feel the same? Do you feel that there has been an impact in science that's affecting the future of how things are done?
Yeah, I agree, of course. We are at the era of system biology, but still, I think we still need some more reductive approach. There is still a lot of things or biological events that are understood by, doing in vitro minimal system, and there is many examples of that. Of course, we need to be aware that there is a lot of biological interaction, but still, we need probably, in fact, strong biochemistry in vitro to understand fully the mechanism. So I think we need both, actually.
I fully agree with you, and I think that we also now have a number of tools that allow us to go into deeper details at the structural level. So like cryo-electron microscopy and cryotomography, which really allow us to look at the structural details at the cell level within the cell. And so I think biology can cover really a spectrum that goes from the precise functioning of proteins to the system, and this is a dramatic change.
Nevan, I believe that one of your favorites phrases is systems to structure. How would you chime in about what Christophe and Bruno just said concerning cryo-EM and maybe biochemistry?
Yeah, no, I'm in total agreement, and the answer is you need both. I mean, several decades ago, it was real, biology, microbiology was really a reductionist approach. You would take one protein, maybe a couple proteins, and study them in a test tube and in isolation, which has its great value. But with the advent of systems biology, one can take a few steps back and globally scan a system and identify connections that nobody could have hypothesized, right? And then you identify a new factor or a new connection, then you can go into a more in vitro detailed system. So the challenge is, as you guys are talking about, is to go back and forth between these large scale unbiased views and these more detailed mechanistic, biophysical, biochemical, and often structural views. That's where the power is. And this is where Jacqueline's saying systems to structure, and then I say structure to systems. So you really want to be going back and forth between these viewpoints. And I would also add that doing so allows you not just to make connections with in the cell or in a biological sense, but allows you to make connections between different scientists as well, because some people are experts on more detailed mechanistic view, others like to look at the larger view. And so you can bring different scientists with different viewpoints together, which I think is going to be touched upon later in this hour. But that's a big part of it too. And if you look at things differently, you bring different kind of approaches together, you can go even so much further together than you can alone.
We have another question on topic from Adam Craig in the audience. Has studying differences in infection of different tissues or differences in antibody repertoire between tissues shed light on how some cell signaling pathways differ across cells? Which one of you would like to take this one? Nevan?
That's for Shaeri, and that's a Shaeri question.
Shaeri, would you like to take this? Congratulations, Adam, for sending it.
I think Nevan is making fun of me because I don't really study tissues at all. So, but sure. I mean, I think that obviously, studying differences in different tissues sheds a lot of light, and because signaling, you can think of it like as at various levels, right? Like, so there's paracrine, there's endocrine. Like sometimes cells communicate just the ones that are next to each other, and sometimes the communication comes from the brain. So yeah, for sure that makes a lot of sense. But I am a reductionist biologist. I try to understand molecules and what they do, and I don't have a big understanding of the big network of on the organism level. But what I can say is that it's fascinating to me how when you do a very solid, you feel study of cells that are in isolation and not in the context of tissue, and then the reviewer says, well, you don't have an animal model, so we don't believe this. And so it's definitely out there. People are sort of interested in knowing like how. Yes, you have shown us a very good phenotype in a cell culture, but how does I don't understand how the cell, these same cells, when they're even in an organoid form, right? Not in a flat, cell culture, but in an organoid form. And Melanie Ott has shown beautifully, like how, hepatitis C, differs when it's in context. So cell signaling is definitely very different when they're next to cells that they should be. But at the same time, I have to also stress that many fundamental discoveries have been made when cells have been studied in isolation. So you cannot completely rule that out, but it would be nice to collaborate with people who work with, tissue models and then see if the same holds true. So, yeah.
Christophe?
So one thing that has been done with pathogens is, one striking thing is that these pathogens often have an ability to colonize a vast number of niches. So, tissues here are different niches. And a number of pathogens are also commensal. So they are able to alternate, live in our body as commensals. So everything is okay. And then for different reasons, because we become immunocompromised, they become pathogens. And so they can, according to the status of our microbiota, the status of our immune system, they will be able to, invade different type of niches, and they are able to adapt to these different niches. And so this is something that is emerging for fungal pathogens, but I'm pretty sure the same applies to bacterial pathogens. I'm thinking, for instance, of Staph aureus, for instance, which is able to infect a variety of body sites. So I think this is something that is emerging from looking at the different types of infection. So the plasticity of pathogens, their ability to adapt to a multitude of niches, and this is why makes them sometimes very difficult to combat.
And maybe I would just add one thing as well, in terms of the science that we're involved in. So we're generating networks, and we often generate these, physical or genetic interaction networks in kind of workhorse cell systems like HEC293 cells or Jurkat cells. But when you go from cell type to cell type, you see differences. You actually sometimes see big differences. We're actually doing this now in the same cell type, plus and minus cancer, and we're seeing really big differences. So I think it is a key point is, it's exciting that there's these differences, but you got to recognize as you go from cell type to cell type, you're going to see different signaling events. You're going to see different networks. And as you go from healthy to disease, you're going to see in many cases, massive rewiring as well. So I think it's really important to look across many different cell types and then ultimately test your hypotheses, and hopefully in an animal model as well, where all the tissues are there and all the organs are there as well. So I think it's a very important question.
So switching just a little bit more towards questions about treatment, are some antibiotics, given everything we've been discussing here and the commonalities with bacteria and such, are some antibiotics promising drugs for treating cancer? Bruno, is this something you could chime in on?
Yeah, the antibiotic had been used for a long time to treat cancer. But the problem, I think, it's maybe more complicated than that, because as you know, there is several papers and many papers now showing that the microbiota is very important for in particular for potentiating immunotherapy. So it may be, I think it's, reading some review about the use of antibiotic to treat cancer, I think, this new discovery that the crucial importance of the microbiota for the immune response against cancer, it's something that need to be taken into consideration from this.
Christophe, did you have something to add?
Well, so I think, the role of the microbiota is very important in cancer, and in that sense, the impact of antimicrobials on cancer will be obvious. I think there are also evidences that the microbiota is important to somehow potentiate some drugs other than immunotherapy, and that are used in cancer. I think we can extend that to neurological disease. So there are evidences that the microbiota has an impact on, say, schizophrenia, for instance, or other examples of neurological disease. So this connection between, in that case, we are not talking about pathogens, we are talking about common salts and chronic disease such as cancer or neurological disease is very important.
So based on what you're all saying, does it make sense then that at a molecular level, there's overlap across different disease areas?
Yeah, it does make sense if you take a few steps back and look at the big picture. As we look across, these different diseases, you see, as we're discussing, the same genes mutated in cancer that are being hijacked by viruses, by HIV, for example, or SARS-CoV-2, or you see the same genes being mutated in neurodegenerative disease that are being hijacked by Zika. And of course, Zika has been connected to microcephaly. And to me, it does make sense. You have these Achilles heels of the cell, they become mutated, and they result in disease X, Y, or Z, or a virus has evolved to attack those important nodes And viruses are very smart. They're not going to waste their time on genes or proteins that are not important or the most important to a particular cell type. So to me, it does make sense, and even more reason to look across these different disease areas, and hopefully these unbiased ways, because there may be some treatment, as we discussed before, for disease X that may even be better for disease Y. And we're starting to see that now with some of these examples with SARS-CoV-2 as we start to look for drug repurposing in a very expedited way. So I'd like to see, more of those, going more in that direction to find those commonalities, because it does make sense. To me, it does make sense at a molecular level. And we don't see it because science is so siloed. Someone who works on cancer doesn't work on neurodegenerative disease. Someone who works on HIV really doesn't even maybe work on SARS-CoV-2. So looking across these disease areas is really important to try to find these commonalities, because it has great therapeutic value, in my opinion.
Christophe?
I think SARS-CoV-2 in that sense will be very interesting, because it's primarily a respiratory infection, but it's also a disease that is affecting the neurological system with ageusia and anosmia. And so understanding how SARS-CoV-2 is impacting our neuronal system is going to and how this is related to the interaction of SARS-CoV-2 with lung cells is going to be very important. So I think, in terms of understanding these kind of relationships, SARS-CoV-2 has value.
Shaeri?
Yeah, so, historically, maybe 100 years ago, infectious disease was most probably the leading cause of deaths. And then what happened was we discovered a whole bunch of antibiotics, and we thought we've got this under control. And then the shift focused on cancer and on, cardiovascular diseases and all the others. And what I got started at least worrying about was that the Western world, which, holds most of the research and development tools and the money to do research, for them, the infectious disease then didn't become as much of a threat. And so a lot of focus on studies and efforts to, have therapeutics went on, towards the other side, right? More the other diseases. And I think what SARS-CoV-2 has taught us is that we should be prepared for, what's to come, not start as the problem is hitting us, but what Nevan has said multiple times, SARS-CoV-2 2020, but, what about 2024? What about, what's coming? And I think that this idea of common, sort of nodalities also gives us more hope that if you actually continue studying these infectious diseases, because they are so limited and because they know the inner workings of the cell, they hit the key proteins in the host cell, we, not only give ourselves a chance to get better prepared, but also, it's not taking away from your cancer research. It's not taking away from any research. Most likely, it will end up, giving us some really brilliant insights into basic biology that will be helpful.
I wonder, Bruno, if you might chime in and thoughts on is the physical basis of pathologies, for example, mechanotransduction, still underestimated, and what are the possible therapies according to you?
So just to come back to what we said before. So we have been talking about common biochemical mechanism, but I think that there is also is fascinating studies, some physical processes that are common to living system, and one which is very interesting is so-called liquid-liquid phase separation that allows the formation of membraneless compartment in the cells. And so this, there are many studies that show that, for example, P granule or nucleolus or lipid droplets are formed by liquid-liquid phase separation, which is a pure physical process. And I have seen a very interesting paper recently in Science showing that lipid droplets, that was not expected, as an antimicrobial activity. So lipid droplets could be part of the immune response of cells against bacteria. So I find this is the new connection between bases. Yes, so mechanotransduction is also another important physical process because, as you know, cells and tissue continuously face mechanical stress, and so there are many studies for the moment that underlie the importance of the mechanotransduction for many cell process and signaling pathway. But so far, but it will take some years. There is no real tradition of what we know about mechanotransduction and transferring to clinics. But I am sure that this will come in a few years.
Thank you. We have another question from MB in the audience, to any which one of you. On the clinical side, can the identification of common pathways for different pathologies open the door to important amounts of drug repurposing in the future?
Yeah, I guess we touched upon that previously. There's not a ton of examples of drug repurposing that are out there for different disease areas. And the question is it because it won't work or because people haven't looked? And I personally think it's more of the latter. And oftentimes, companies that have a certain drugs, if others find another purpose for them, it's other companies specifically, it's not financially beneficial for them to kind of push in that direction because the patent is owned by another company, and anything that they find and apply would be attributed to the initial company that has the patent on the drug, which isn't a good reason not to push in that direction. But the fact that you do find all these commonalities, which is what we're talking about, and they're starting to see some good examples with SARS-CoV-2. When you're pressured to come up with something, when you have nothing else, we're starting to see some good examples. Well, remdesivir, which is pretty much the only drug right now, antiviral drug that's being used for SARS-CoV-2, was initially for Ebola, right? So that's an example of drug repurposing. And there's a number of other drugs that are now going to be coming online that are hitting host factors that have been developed obviously for other diseases, because we didn't know about COVID until this year. I think there are going to be more examples in the very near future that are going to prove fruitful with respect to drug repurposing. And then I think that will hopefully induce others to go and take a few steps back and look at, other diseases and other drugs and explore the possibility that certain drugs could be used for other diseases. Because if it's the same gene or the same protein or the same complex or the same pathway that's being mutated in disease X versus disease Y, if you have a drug that's having an effect on that pathway for one disease, why wouldn't it work on the other disease if it's the same set of genes or proteins that are being mutated? So I hope that the SARS-CoV-2 pandemic, which while it's been very negative, one of the positives will be pushing scientists to look more at drug repurposing as a valuable tool for treating disease in the future.
So beyond drug repurposing, do you all believe that the pandemic has changed research in a way that would allow more easily for the identification of commonalities across disease states or disease areas? Say beyond the repurposing of drugs, do you feel that the pandemic has actually affected research in science?
I have thoughts. Can I start, guys? So, I think, for us to find these commonalities and to find potential novel therapies for disease across different disease areas, one needs to increase communication and across different scientists, different types of scientists, different scientists working on different disease areas. And I think what we've seen in this pandemic is the increase of communication and collaboration across many different entities, not just between different academic institutions, but between academia and pharmaceutical companies in I would argue, a very unprecedented way. So yeah, I do believe that the pandemic is teaching us how to work better together and more quickly together, which almost certainly is going to identify these commonalities and point to new therapeutic directions that we wouldn't have thought about. So it's really, in my mind, about communication and collaboration, and that's what's happening now more than ever. And hopefully the challenge is to keep that type of spirit and infrastructure in place. So as Shaeri was saying, so we'll be ready for the next pandemic, or let's just study other diseases in a similar fashion.
Bruno, what are your thoughts on this? Do you feel like the pandemic has changed the way we do?
No, I fully agree. I mean, I think that, if you look at what the virus do inside the cell, so it's just incredible. I mean, it seems to affect, so many functions. So if you want to understand or to have at least a clear picture of what is going on, you need to understand several processes, not only trafficking, right? If you look at that, so it's I think it's a good example of what can be done, and also the fact that so many people are concerned about this research. Of course, I think it's can be an important point for the future, and to foster collaboration between different disciplines.
Shaeri?
Yeah, I want to say something on a lighter note. Like, Bruno has always been a scientific hero for me on his work on RABs, and the highlight of my day one day was when I received an email from Bruno asking about collaborations on SARS-CoV-2. That was so awesome. I know SARS-CoV-2 is bad for everyone, but that was the reason why I could even come to that stage where I could possibly think of collaborating with someone like Bruno. So I think it has definitely opened up a lot of avenues for collaboration across continents. It's not just about, okay, who can I find next to me that can help me do this quickly. It's now no one is traveling, so the whole world is a one lab, so we can all do things together and share it on Zoom. So I think that is definitely a big positive to come out of this pandemic.
Christophe?
Well, I agree with Bruno, Shaeri, and Nevan. I think the pandemic has definitely changed the way we communicate. I'd like to point this example of the collaboration between QBI and Pasteur. This all started by a phone call from Nevan, I think in March, something like that. And so we decided to collaborate. And so the team that has been involved at Pasteur is a team of Marco Vignuzzi. It's been highly productive, but Nevan and Marco have never met except by Zoom. So that's totally that's a complete change in the way we do science. And I think this is, it's not going to be limited to these pandemics. I think it's going to really change things. We now know that we do not have to fly to be together, and that's good for the Earth. And this is going to change our way of working. Well, we still need to meet from time to time, because moving to San Francisco for good food is a good thing to do as well.
Well, and let me add to that. I agree. Someone told me, a colleague told me recently, that Zoom is great for carrying on relationships that have already been established, but it is kind of a challenge to initiate new ones. And well, Marco and we had obviously just met via Zoom. QBI and the Pasteur, as Christophe's alluding to, for the last several years have been working together, building up a relationship, just like we're doing with the Curie Institute and Bruno, through symposium, getting scientists together. So there's something to be said for still getting people together. I think there's a creativity that is being lost because we're not together, and that we have to do the Zoom all the time. So there's pluses and minuses. So when COVID-19 goes away, I do agree, Christophe, that we'll be doing more Zooming, but I don't think we can completely get rid of the in-person symposium. And partly I say that because you guys were supposed to host the next symposium, Christophe, at the Pasteur QBI. So when we want to come out there, the food is much better in Paris than in San Francisco, I would argue.
I just wanted to add that, yes, there have been absolutely phenomenal collaborations and connections among labs across the entire planet, as we've seen. but also unusual collaborators have popped out. So in our case, the French Consulate of San Francisco has been a fantastic partner for us, and also facilitated a lot of what has been going on with the research, because our drugs and compounds that were going to the Institut Pasteur at the beginning were blocked due to travel bans and all kinds of things, and the consulate stepped in. And so science has also brought in all these other entities to the table that are working together, which I think is rather incredible and incredibly important as well. But one question I had for all of you is, this is all sounding really fantastic, but what can we further do to facilitate collaborations more effectively to allow for the identification of these commonalities? It's working incredibly well in the context of COVID-19 right now, but eventually, I suppose, we all hope we're going to get out of this in about a year or so. Then what? How do we continue facilitating these collaborations? What are your thoughts?
Maybe I'll start by saying money. I think money is the greatest kind of collaborative facilitator. So having these agencies like NIH, and I think we should have NIH partner more with agencies in other countries to really encourage these cross-country collaborative efforts, which have proven to be so fruitful in this pandemic. So I really want to see more of that. And then a lot of the stuff that, to be perfectly honest, what we've been doing in the last few years is getting people together through creative ways, through symposia, and you don't need a lot of money, just a little bit of money to kind of grease the wheels with certain collaborations. And this is what we've been trying to do here, with a little bit of money to get people connected, and then that leads to much bigger things in the future.
Yeah, I fully agree with that.
I wanted to add that, I really feel for our junior trainees, especially, postdocs who were planning to apply for jobs and their independent careers, and SARS-CoV-2 hit, and, hiring stopped in a lot of different universities, and we have to protect these people and their careers, and definitely what Nevan said, money is important, to give them grants, to give them more time. There's a backlog of hiring, and so we don't want to lose this generation because of what happened this year, and perhaps it will, move into next year. So I really want to see some genuine efforts on the behalf of these governments to actually protect, some students that are on the verge of graduating, or postdocs that are on the verge of getting a job, to help them somehow, start, their own careers, independent careers as well.
Christophe, any thoughts?
Well, obviously, money is key. I think Nevan mentioned NIH. I think NIH is actually making efforts to support groups in Europe, for instance, and worldwide. I think the same should apply to European agencies, and I think this is very important. So Europe has started funding some other countries, but clearly programs that would put teams from different continents on the same level would be a good idea to go. I think what is also key is to promote our ability to brainstorm, to exchange ideas. I think this is something we've done 3 or 4 weeks ago. And this has been very important to try to, stimulate our interactions between QBI and Pasteur. And I guess you've done the same with other institutes. And I think this is something that is important. This is why I agree with you, Nevan. We need to, meet. We need to resume meetings, because this is definitely where the brainstorming is the most efficient, when we meet in person.
Bruno, any last words? We're coming to our last minute, so I'll ask all of you to have a few last words.
No, I was just thinking, so it's true that, money is important, but as Nevan said, we don't need a lot of money at the beginning, actually. It's, we did that inside Curie, or within this QLife Institute. I mean, you just give a small amount of money to initiate a project, and then people start to speak to each other, and then they can apply to bigger grants. So I think it's very important to have that in mind.
Shaeri?
Yeah, I completely agree with everyone. Yeah, money is not like running out or anything, but it would be nice to have some small seed money to start a collaboration.
All right, so unfortunately, we've come to the end of the hour, and it does seem like the audience has more questions. So I encourage all of you to answer in written form after we close. And any last words from any of you?
Maybe I could just chime in. I mean, just to reiterate kind of a higher level message. I mean, clearly this has been a tragedy, this pandemic, but there are a number of positives. And for me, it's seeing really the power of collaboration. I knew it was important, but I've just seen how important it's been over the last 6 or 7 months. So I got to say, I am so excited about the future. You know, when this pandemic goes away, there's going to be science on steroids, in my opinion. This is what I want to see. And it's so great to see science front and center on the newspapers and in the press instead of, you know, baseball or football players. Nothing wrong with that. I'm a big sports fan. But I think people have learned a lot about science in general. And the science is so bright. Obviously, we've got several months painful ahead of us right now. But, you know, as Jacqueline says, a year from now, things are going to be looking a lot better. And I'm so excited about the future and what science can do for all of us.
All right. Well, thank you very much, everyone, for joining this panel. It was wonderful. And I know we will all be in touch and continuing with our collaborations. And with this, we're going to end on our hour. Thank you.
Thank you.
Thank you very much. Bye-bye.